This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
Kia ora and welcome to the OI Podcast, the podcast from the Otago Institute for the Arts and Sciences as well as scientists. The Otago branch of Te Apianga, the Royal Society of New Zealand, where we meet and chat fascinating people from the worlds of arts, sciences and Mataranga Maori. This is brought to you by OAR Otago, Otago Access Radio on 105.4 FM or wherever you download your podcasts from, such as Spotify, Apple, I don't know where else people get podcasts from. I just search on the internet. I'm just a bit of a generalist when it comes to that. So anyway, I'm Trudy, and although Dr. Barb is in the country, my co-host, so welcome back, Barb, she's brought a bit of a bug back with her. And that's really interesting because bugs is the topic for today, but not those kind of bugs. But she's brought something back and she doesn't want to share with us, which is very nice of her. So I hope you're resting up, Barb, and lots of fluids and... yeah, and hopefully we'll see you next week. But today I have with me via Zoom Aimee van der Reis and Richard O'Rorke, both doctors. Is that correct? You both, yeah, done that?
Yep,
PhD, yeah. Good on you. Got a Scott about that. It's a lot of hard work. From the University of Auckland. Sorry?
It's a lot of fun. Don't
a lot
put
of
people
fun.
off.
Is it?
It's really worth doing. Yeah, I think so.
Oh,
Very
cool.
important.
Why is it? Oh, well, anyway, they're from the University of Auckland, the School of Biological Sciences. So how did you both end up there? Aimee,
We both did marine science degrees.
Yeah.
Yeah, so I went there to do a PhD. Aimee, did you go PhD level or were you there beforehand?
No, I was there to do my PhD as well.
Mm.
In my Auckland, like we've got a marine science department down here in Otago, being very provocative. Yeah.
Yeah, you've got an amazing, Otago's got really great facilities and is doing some really great stuff in the spaces that we work in as well. I think it's true across all of the universities of Aotearoa that they're doing great stuff. For me, it was just proximity to people I knew and where I grew up, and also just one of the reasons is that I was interested in using DNA techniques in a similar way to what microbiologists had been doing. I had a background in molecular microbiology, and just an opportunity came up at Lee Marine Research Centre to work with Andrew Jeffs looking at, and Shane Lavery to look at the diets of Phyllosomas, so the larvae of lobsters or crayfish when they migrate hundreds of kilometers offshore.
Oh,
and
cool. Yeah.
And so it was really just that at that point, a lot of people were very skeptical about whether these methods could work, and just having somebody or a couple of people that were interested in finding out what would happen if we applied these techniques and whether they'd be successful was that exactly slotted in with my interest. So that was more than 15 years ago. But at that time there were very few publications on it, so I was really grateful to have that opportunity to do something that I was interested in.
Fantastic. And for you, Aimee, yours is a bit of a longer journey. Is it like kilometre wise?
I started my PhD. I'm from South Africa, and I decided to do a PhD, having done my undergrad and honours in South Africa. So I guess just when you're looking through what interests you, I've got quite a solid background in more the molecular and genetic side of things. And I always just wanted to go into the marine side. And so I was just looking at different options of what projects were available. And it's with the same supervisor Richard had, and that's actually how we came to know each other, but with Andrew Jeffs and Shane Lavery. And I was working with it was a project that was co-led by Cawthorne, looking at scampi, so the deep sea lobsters, looking at diet and population structure. So also very interesting and good fun. And it was just a project that enabled me to up my skills in the areas that interested me. So
fantastic. So I suppose the key message there is if you want to do a PhD, it's got to be fun. So it has to be something you're interested in and working in finding something where you can contribute as well as learning a lot too, like widening that broader science out there in the community. Yeah. Cool.
Yeah, no, definitely. It has to interest you. Otherwise, it's a long time and a lot of effort.
Yes, yeah.
So,
and you, Richard, you sort of were at the start of something. So, if you said there weren't any publications or something you could get into the ground on.
Yeah, no, at that point, there were a few people looking at using PCR techniques in particular to explore the diets of animals. There was a paper that no one ever cites, but it's from the late 90s by someone called Oberholster, who sort of thought that maybe it'd be really interesting to use zooplankton as a way to get a sort of an idea about low abundance algae by looking in their gut contents using PCR-based techniques. And I read that paper and I thought, wow, that's quite exciting. And so I sort of was looking around for other sort of, well, for where I could look at crustacean diet. I was also interested in looking at virology too, but it just happened that out of the two, that opportunity came up, and that's the course that I ended up taking. So yeah.
I suppose it's not that much different, just in scale, than looking at middens or old rubbish tips from human habitation to see what we ate and what happened and that kind of thing. It's just on a different level, yeah.
Yeah, and there's a lot of overlapping techniques. In particular, you're looking at just like sort of with ancient or historic DNA that'll be degraded through time, and like with stuff that's passage through the gut system, that's just going to be degraded by digestion. So you have a lot of overlapping concepts that are involved. And again, with this project that Aimee and I are working on at the moment, the DNA that ends up on the exterior of a car has a pretty rough time. So we have to use some of the same ideas too.
So perhaps we should talk about that, the DNADRV or DNA drive. Like when you look at those letters together, you think, what on earth is that? But when you it's a bit like reading mathematical formulas. If you read them out loud, it kind of then makes sense. So tell us a bit of an overview. Yeah.
Well, it's not just like reading a mathematical formula. It's also just like puzzling at an intersection where you look at a car number plate.
Exactly, yeah.
You sort of like wonder what's that saying? And, like to use a corny sort of bridge, that's kind of what we're doing is we're asking what can number plates tell us about the ecosystems that people drive through and are special to people. And we just thought that we could ask members of the public as well as people who we know frequently drive out into the countryside if they could provide us with swabs of the surface of their car number plates. And from these, we can extract DNA and RNA, and we can then identify the organisms that the cars smash into. And we all know from personal experience that we do collect quite a lot of insect material, but we're also looking at other organisms that might be within the guts of the insects or that they're interacting with when we collect our samples.
So are you interested in the ecosystems or the bugs or both, is it?
No, we're definitely interested in both. I think Richard and I we like collecting data and not just a single type of data. We want all the data, and then we'll worry later down the line about how we're going to answer all the questions that we have. But the main focus is insects, but we are including to look at, as Richard mentioned, for example, what's in the gut of the insects. So we've got certain gene regions that we can use to look at plants, and we've got certain gene regions we can use to look at bacteria. And so later down the line, it's just going to be interesting to look at the ecosystem as a whole. So what's happening in this food web of certain areas. But that's the big thing at the moment is we want those insects.
So how, like, bugs are pretty small, and when they end up on your number plate, they're even smaller. So how do you get the actual DNA out of the bug and then out of its stomach? Like, I wouldn't even know where to look. Yep. Or do you just melt them down or something?
Well, pretty much. Like, the thing is, there's an enormous amount of DNA in in an animal, even a small insect, each of its cell has a nucleus that has all those kind of gene regions that are important to making it function. But then also we're looking at mitochondria, which there's a sort of an order of magnitude or more of again. So there's just so much DNA packaged up inside organisms. And that's why a lot of these techniques can work, because it's even a very efficient digestive system or a very efficient degradation of DNA from the heat and whatever bits of weather happen to a car number plate, there is still going to be some DNA that survives. And through just careful application of methods, we can access that. So we essentially take that swab of the front of the car number plate, we put it through a series of extraction procedures to purify the nucleic acids. And then at the moment, we're using a PCR-based technique just because it's the most cost-effective and tried and true approach where you can really isolate these gene regions that we know are very informative about what species we might be encountering. And then we amplify those up and we throw them in a sequencer in bulk.
Right. Sounds like a lot of fun, actually. Feel a bit like Frankenstein in the lab, seeing what the secrets are in there and in the DNA. Yeah.
Yeah, and there's a lot of pleasure in refining these techniques and just getting them better and better as we go along.
And I was going to say, because it's a repetitive study, each time you do something, you might find ways of improving the technique for getting the DNA, but also about the study. Do you find that you've been able to refine how you get the bugs or?
Yeah, definitely. So we did a preliminary study in 2020, in the beginning of 2024, and that was pretty much the start point of our refinement, and it's continued. And it's really just an iterative way of trying to do things. So we had this preliminary study, then we did another one later in the year, towards the end of the year last year, and that incorporated some of the improvements that we thought the first round could be better at. And then after doing that, we're like, well, if we go wider, this is what we need. And a lot's actually also come down to us getting public feedback.
Right.
And it's not always just in the lab. Sometimes it's things about the kits that we ask people to use. So it's as simple as essentially having a place to write notes about your journey so that you're not having to remember to fill it online later. So that's not just the lab techniques. It's also just the feedback that's really helped us from the public.
I have to say, this is one project I know a wee bit about, but in case there's somebody out there who doesn't know about it, talk a wee bit about what the kits and how they work and what you want or would like people to do.
So we're asking the general public and anybody that's really interested, perhaps what's happening in the environment around them and the insects in the area. The idea behind the project is to look at insects. As we've already mentioned, maybe looking at the wider ecosystem and food webs as well. But we know a bit about some insects, but there's also so many we don't know anything about. And so we're asking people to go about their day as normal, but if they can perhaps, before they drive from point A to point B, if they can clean their number plate before they go on their drive. And there's a little alcohol pad in these kits, as well as washers for your number plate. And then you wipe down your number plate with that alcohol pad, and that gives us a background noise of if there's any sort of remnant DNA that's on your number plate. So it gives us some baseline data. Then you'd go on your drive, hopefully collide with some insects along the way. And then at the end of your journey, we provide a little insect tube. And any insects, you could try flick them off. Sometimes if it's very windy, it's quite difficult. So don't feel too bad if you flick an insect off and it flies away with the wind.
Do it in the garage, perhaps. Do it in your garage if you got one. Yeah.
Yes. Well, it's not always possible to obviously if you're driving from A to B and you're going to sample at B, there might not be a garage,
Exactly.
but yes. So that does happen. But the idea behind that is that if there's a whole bunch of insects on the plate, we want to try remove the insects to remove a lot of the insect DNA that might then in a way overwhelm the signals that we get, because we also want the insects that hit the number plate but bounce off. So for example, your bigger insects like your wasps or perhaps your bees, because they're not going to necessarily stick onto the number plate, whereas your smaller midges will. And so by removing that, we can get these trace signals of those insects that collided but didn't stick. And from there, once you've removed any visible insects, you take another alcohol pad, and you wipe down the number plate again, and you pop it in a tube, and that's it. So quick and easy.
And there's lots of drop-off places, and we've got a link to the DNADRV website, and that's got a map. So that's really interesting. So a bumblebee or a wasp can collide, not stick, but you can still get DNA from the trace they leave behind.
Yes, so I think that's really what this technique is. Even though DNA has come about, obviously any of the methods a very long way, a lot of it's based on forensics. And so when you think about forensics at a crime scene or something like that, there might not really be that much DNA around. And so any sort of trace DNA, and because we're using a technique that amplifies the DNA, and those set gene regions, it means that we can make a tiny little bit of DNA
go
a lot.
Long.
Yes.
That means Hollywood's going to have to rewrite a whole genre of detective movies, because, nobody can go and clean a place anymore because there will be traces of you no matter where you go. It's food for thought. So then thinking about the insects, we know a lot about some insects and we don't know very much about a whole lot. Is that what we're trying to achieve here?
Yeah, so this is what we've found. I'd have to emphasize that neither Aimee and I are entomologists. I can barely say the word, let alone do it. But
we have had them on the show, so that's all good. They'll cover that bit. Yeah.
And so we are working with teams of entomologists, and they are helping us out with morphological identification, but also just telling us a little bit about the life history of things that we encounter as we sequence people's samples. However, so on that pilot study that Aimee mentioned, all these cars, it was actually a vintage car club.
Oh, so yes, that's on you.
The car club. And so they were all driving the same route. And what we found is that all of them were encountering something called a black-winged fungus gnat. Oh. And the thing about these is that they seem to have quite a crucial ecological role. We've been talking to people at places like PG Wrightson's about how we theorize their importance, but and particularly to farming and sort of sustaining predatory insects. But we also have just found through looking through the literature that they're hardly ever mentioned. There's been as far as I can find, 2 studies, one done over 100 years ago characterizing them, and then one done more recently in the last decade. And so there's this really sporadic interest in these massively abundant, both numerically and in terms of biomass abundant species that must be having a really profound impact on our ecosystem. And if they disappear or change, they will
have an impact.
Yeah. And what we want is some sort of baseline where people can look at what's happened and think like, oh, if only we had some information from a decade ago or further ago, which is what we lack now. But hopefully going forward, this study will provide that basis of saying like, well, what's changed and how has it changed?
So how long do you think you need to go, or like we'll just keep going until somebody says, no, there's no more money, or,
Yeah, so I think that we're going to try and sustain this project in some form or another for as long as we possibly can. it will be a sort of feast and famine
Right.
scenario where if we get funding, we'll be able to do projects such as what we're running over the summer, where we're just inviting members of the public as well as other sort of organizations to just submit samples to us, and we'll sequence the samples and tell them what we find. We won't be able to do that after March because we won't have any funding, but we're going to just look for opportunities where we can collect samples and continue to do it, and also to look for other funding opportunities. We're also working or looking to work with schools because they already have this wonderful infrastructure in place. And also we've already received a lot of really great feedback and samples from schools. And through working with teachers, students, parents, we think that maybe this could be a really good system that's already in place that we can mutually benefit from. Students and teachers and parents can learn about this methodology, and learning about genetics is benefit everyone. You don't have to necessarily want to become a scientist. Just being fluent in some of the concepts is
Right.
going to really help you in the future. And also, we can benefit from the fact that if people are wanting to collect samples from their community and to understand what's happening ecologically in their area, we can then start building this database that'll hopefully help future-proof some of our ecosystem resilience.
Yeah, I was going to say with children, you're going to have each year you get a new crop, so to speak, that'll come through that could, contribute to the program. So it's not like you've got to go out and find new people to take samples each year. And I think that community basis would be really interesting for people because they do like to understand what's going on in the community and what's happening. So that would be, I think that's quite smart. I wonder if the Scouts or whatever have considered a bug collector's badge or something. This could be part of it. So I think there's lots of opportunities out there.
Yeah, we have had interaction with the rangers.
Oh, yeah.
Yeah. But I think that would be really nice if we could get things like that happening. And essentially I would like this to be a community project where we ask people what's important to them, how do they want to take samples, and then we'll work with them to provide what they want and how they want it done. There has to be some sort of structure in it because Aimee and I need to sort of be able to do the sort of sequencing work. But within the certain guidelines, I think that there's a lot of scope for communities to decide how, or people to decide what they want to know about and how they want to do things.
So looking at what you've collected so far, are there any gaps? Like, for example, my husband's a shift worker, so he always leaves home at 4 PM and comes back at 2 AM, and I must really talk him into doing it because I'm not sure that, do you have samples from that kind of time range? Are there areas of time of the day or areas of the community that you think could do with a bit more enthusiasm into collecting bugs? Yeah.
We've had a lot of people really be enthusiastic about the project and actually emailed us and asked us what areas are we missing, what times should they actually be doing the sampling in. But the idea behind the project is that people need to do it at their convenience.
Right.
We'd rather provide people more kits and get more samples. And they're welcome to sort of, if they're interested, to sample during the day and then also sample during the night and see the difference. Like, that's just more data for us. We're not going to say no.
Sorry, on the kits you put the time, don't you? The time and date. So when you put it in, you can have a look and see. So I suppose in some ways you can, if you want to compare apples with apples, you can from this time period last month to last year or whatever. Yeah.
Yeah. So we have some people that are much more structured in how they prefer to do the sampling, but then we also have some people that just want to give it a try, and of course they must do it in their convenience. But regarding the gaps, we don't know just yet because it's relatively sporadic because you're working with anybody that's interested. Probably nighttime would be quite a bit less, but we do, now that we're getting into summer, we do have a bit of more of a shift, sort of the dusk into night,
right?
Because people are out and about, which is great because that sort of aligns with insects also being out and about, given now they're coming out of their little cycle. So.
Oh, that might answer my own question, because when I did it last year, I thought, oh, there aren't very many bugs on, like, even just driving at night, I don't see as many bugs as what I did previously, but that's just a casual observation. I could be at the wrong time of the year, or, climate or weather. Does that impact when the bugs are around?
So this is what we're finding, is that when we work with people, there is this natural tendency to throw out hypotheses about what insects are doing just based on doing it once or twice. And then we're hoping that as we accumulate more samples, we can address some of these hypotheses. So we are hearing from people that say, like, I used to drive this route 10 years ago and I used to encounter more bugs. Or, like people saying, I was really surprised I didn't encounter any bugs. Was it perhaps the time of year or the time of day that I was driving at? And just through that natural curiosity naturally sort of lends itself to a scientific hypothesis, and we can help people sort of look at that from a more data-driven way. So, yeah.
So you collect the data in case somebody wants to answer a question.
Yeah, that's the motivation. At the moment, this project is about gathering the data, and then hopefully people can use the data.
Yeah, and we've definitely had some people sort of structure their own type of sampling. So they've gone to different areas and been like, okay, well, I did this, now I'm going to do this. And they tell us about it, which is obviously great. And then they also, as Richard said, they sort of were like, okay, well, it's probably because, I drove really early in the morning, and in comparison to the evening, I had a lot more of the nocturnal insects come out. And so then there was a lot more on the number plate, and I think this also sort of generates a curiosity, but it also enables that wanting to do another sample or try a different time. We've had a lot of people comment that I didn't get any insects on my number plate, and then further on they enter the data again and they're like, finally got some insects this time. And that's really great to see as well, is that people are just becoming, maybe they were already observant, but it's nice to see that people are actually sort of, I guess, more committing to memory what's going on during that period of the day, because they do need to physically go and clean their number plate for us.
And I suppose it could be a good school holiday activity, like when you're going for your drive to picnic to the beach. It'll be something that you would do before you go, and then when you come back, collect bugs, and next time you go somewhere else, you could do something else again. Yeah, you've sort of weaving that into the school holiday activities, yeah.
Absolutely. I think that it can make a long drive with the family a little bit more enjoyable and fun.
Yeah, if you're not going to be quiet, we'll stop and we'll do a bug sample or something like that. Or it could be a motivation to do it.
I think that it's more of a carrot.
Yes, yeah.
But yes.
Yeah. Yeah, nice. So you said something about people can enter their own data, but they have to drop the samples off. Is that?
Yeah, so we have, as well as the physical notes that people would write on the kit themselves, we do encourage people to please enter it online, because that helps us. It also gives us a little bit more information, and people can also add comments. And so they would enter that data, and then they can drop it off at any of the hubs around the country. So yeah, they do enter their data online, and then sometimes if they do put specific comments or anything like that, we do go through them and try to follow up as fast as possible.
So when did this start? Sorry, did you say 2024 was your first?
Yeah.
And how long, or like?
Well, early 2024 was just a preliminary project. So this study this official one. I guess where we involved a lot more people in the community, has gone from March this year, and it ends. It will end March next year.
Okay, so
and it's also sort of escalating, really. So
right.
Sporadically in 2024, and then we've begun this project. But in the recent months, it's really growing in the number of samples we get back. So yeah, and the reason why I'm sitting in a car talking to you now is I've just been out. So I've moved from Taupo Library down to Palmerston North, Central Library and Lower Hutt Community, or War Memorial Library and Patka Porirua Library. And I know this is bad for podcasts, but if you sort of,
Oh,
if I
yeah,
can show
look
you
at the
back
back.
of my car,
There's
it's
all
absolutely
these.
full of all these kits that have been returned to us. So, it's been really amazing just getting this feedback. Working with libraries, so I think are a wonderful hub for communities, and it's a really good way to get things from people and to
And also just gauge a bit more of interest about insects. So we've had libraries do amazing insect displays to promote the
Right.
project
Oh,
as
okay.
well.
Yeah.
And they've gone, some have really gone out their way to make these sort of cardboard insects out of cardboards and like put them up. And of course, along with the display goes books about insects, so engaging with children. But of course, this project is a project for all ages. But yeah, that's just been really, really wonderful working with libraries.
So earlier this year, or might have been last year, we had a chat with Jenny Yent from the Entomological Society, because Dunedin won the Bug of the Year competition this year, and that was also around promoting insects and bugs, and it was quite interesting to see how communities would pick up and get quite interested in bugs. So yeah, I think a wee bit of competition can motivate people to participate. So have you got any results about what areas or regions have produced, sent in the most samples? You've got any results you can share?
We should have that data, but we haven't actually collated it yet. But you're right, we should do something like a top town of who's delivered the most into us. It will be slightly more biased by where Aimee and I started asking for people to set up drop-off points first. It does seem that the longer a drop-off point's been established, the more samples we get from there. But I like this idea of having
regional competitions.
Yes, yeah. We might suffer from small city syndrome down here in the south, but we like to pull above our weight. But yeah, I think communities would be interested to see, and then what percentage of compare our community size. So, yeah, and then. And then, as you said, because people are interested in the bugs and what happens in their community, then you could even then have a table of what bugs come from what communities, and then do it by bug as well. So I never thought I was that interested in bugs and data, but here we go. Yeah,
Yep, and there we go.
So
I feel like being interested in bugs is maybe something that we have when we're like, and everyone says this about certain things that we have when we're younger, but I think that there is a real fascination with these quite unusual, like they're so unlike a mammal and quite often incredibly beautiful, and but yeah, it's just they are quite an enchanting organism, a lot of insects. And I think that we maybe do lose touch with that as we get a bit older and just distracted by other things.
True, probably true, because the other part of the Bug of the Year is they had photos of them, and when you looked at them close up, it was amazing how just beautiful and colourful they were. It's just, I wonder if it's a size thing, because as you get older, you get further away from the ground, and so perhaps you just don't notice them as much. Yeah. So, where you go? Yeah.
No, I was just going to agree with you. And some of the just what look like little black sort of spots on a number plate, when you look at them, they can have these incandescent sort of rainbows sort of shimmering across their bodies. And I think that's another area that Aimee and I are going to look at in the education side of this, is just the looking at side of things, because you can now get such affordable little cameras and adapters for phones and stuff. And I think it just adds that extra level when you can really sort of zoom in and have a look at the bodies of these organisms. Appreciate how beautiful they are.
Because you get these apps, identify this plant, you could get one, identify this dead bug, or something like that. I'm sure you could sell that and fund your program going forward. So, yeah.
Well, our naturalist is obviously really great for that.
Right. Yeah.
So, so I don't think we would go and specifically make app for that or anything, because it's great that we have the taxonomists on our naturalist, like being like, this is definitely it. And that's been a really, really good thing. Well, I guess a nice surprise was one of our collaborators sent us a screenshot of one of the insects that he was identifying. He's specifically an expert in mayflies, and the notes was that the bug had been collected from the DNADRV project. Yeah, and that was very cool to see. But it's a really great way. So if people are actually, if the insect is preserved enough on the number plate, and people would want to know, and big enough, that would be a factor. If you're taking a picture with your phone, then there are people out there who would help you identify as well, if that is something that would interest you.
So that's iNaturalist, isn't it? Yeah.
Yes.
So are there any studies like this elsewhere in the world?
Yeah, there has, for quite a while, since the invention of the car, there has been interest in how you might use it as a sampling tool sporadically.
Yeah.
Which I guess when Aimee and I sort of talked about this and we had the idea, we thought that we were wholly original. And in a way, our project is original because we really benefit from some of the technology that's happening at the moment. But there's been every sort of 5 or 6 years over the last century, there'll be a publication about what's called an auto catcher or some sort of such name where a giant steel frame has been mounted to the top of a small truck or van or even a regular kind of coupe style car, and somebody will just drive around with a net and use it to capture insects. So there's some really charming photos published in scientific journals of these devices, and even quite recently somebody has then taken the insects from such a net and used a genetic approach to identify the insects. I guess that what we're using is not a method that's designed to capture insects. It's designed to capture DNA. So DNA. So I guess the point of difference that we're using is that just by swabbing the surface of a vehicle, you can identify the different insects that have left traces of themselves when they've collided with the plane.
So you don't actually need the whole body; you just need the trace. So yeah.
Absolutely. There's a lot of information in the DNA.
Yeah, and also even though some of those images with the nets on top of the car, they're really they're very cool, but it's not very readily scalable because you're not going to get that many people wanting to put big nets on top of their cars. So that was I guess the design was a little bit unique in the way that at least within New Zealand, your number plates are standardized size. So in terms of science, that is a variable that we could control because everything's the same. Yeah. So we know that those insects they're only being collected from a certain area. So with the variability of makes and models and all of that of cars, we know that it's set, and this is the only area that we ask people to at least clean.
Does it matter that cars are different in height? So while the number plates standard size, they might be different heights from the ground.
So we are collecting data on what the make and model of the car is as well. People provide that, so we can account for that in models. And so it stands to reason because certain insects do tend to be associated with different heights above.
I was going to say, they fly at different.
So absolutely, and we are trying to collect the data that will allow people to account for that. So hopefully it's built into the model. Like, there's no comprehensive sampling method. What we've tried to do is add an auxiliary method that will complement the existing sort of methods, which tend to be what I would call point sampling. They'll be pheromone traps or some other kind of lure that will trap an insect, or a malaise trap, or something that's at a fixed point and it samples what's in the area, whereas a car moves across the globe, and it samples an integrated transect of all the insects that it encounters. So we're not going to get everything, but what we're going to do is we're going to get a different kind of data that can then help improve the resolution of what we're seeing.
It'll be a huge, broad sample of what's out there, rather than just a targeted small area. Yeah. So, insects. What can you tell me about insects? Because you've all done marine biology, aren't shrimpy? Aren't they? Don't they call them the insects of the sea or something?
I think, yeah. So I can't personally tell you much about insects, because I have mostly, well, to the extent that I've looked at any organism, it has been plankton. But I think that, again, going back to sort of talking about what motivated Aimee and I, is that we were used to looking at marine and freshwater systems where there are a lot of these methods that sample integrated areas. There's been a project called the using what's called continuous plankton recorders, which is just like a device that you can drop behind a ship. And these have been for decades to sample plankton across the oceans on vessels that's called vessels of opportunity, just vessels that opt to drag these behind them. So you're getting these nice sort of transects across the globe in certain areas that have high shipping activity, and there's not really been anything like this for terrestrial organisms, like not even like dragging a net behind a ship or anything like that. So it's just thinking about how we can apply methods that have been really successful in the ocean to look at insects and to understand what's happening here.
Fascinating. Yeah, sorry.
Yeah,
Aimee.
but I guess no. I was just going to comment that we're learning a lot about insects at the moment. So yeah, that's actually probably been quite refreshing. It's like it's gone from marine a bit to terrestrial, and we're certainly learning a lot that we didn't know, and learning how much people, at least as scientists in the literature, that we don't know. So that's also really nice.
So it gives you quite a strength, some more to your personal scientific development as much as the communities in the scientific world. So that must be really quite fascinating. So
yeah, and it's nice that we can add to the knowledge base and just as mentioned, just create this sort of baseline empirical data to in future know potentially what's caused some changes that might be noticeable later down the line.
And you mentioned, was it PG Wrightson's before, or was it one of the farming groups? Yeah. So yeah, how did that interaction come about, and what were they hoping that we might be able to help them with? Using the royal we there, like I'm just hanging on.
Yeah. So that was just from somebody seeing us when we did a television appearance and thinking like, wow, this could be really relevant to the farming communities.
Yeah.
Obviously, insects are very important as pollinators and how they interact with crops, and so finding diverse ways to sample insects is really beneficial to primary industries, as well as just being something that we're a bit curious of and concerned about. So that's how that came about. And yeah, we've had a few meetings, and I sort of hope that we can develop things with them as well.
Yeah, I think it's kind of another tool for the primary industry to look at what, bugs or the opposite pollinators, because one man's pollinator might be another person's pest. So, it's about that. Yeah.
Yeah. And the thing about this method is it's a bit agnostic. We don't we just sort of happen to sample whatever it is randomly counted by a vehicle, whereas a lot of agricultural sampling is quite targeted. There'll be certain pest species maybe that they're interested in, certain pollinator species they're interested in, but like you say, like a pest species might actually be a very important pollinator species.
But what's nice is just also working with all these different groups is that there's a lot of rich knowledge that's never been put into some sort of written or searchable, findable form. And so when you talk with the different people, there's just so much more you can learn from them. And so that's also been really, really great. And it's just like you can tap into people's sort of expertise about different things, and just learn a lot more what's not actually in the literature or online.
So I was just thinking that it must be quite fascinating because it's quite an open, broad study, so you're not really constrained down one route. It'd be quite easy, or relatively easy, to change something if it wasn't you've done when you've reviewed, how to get your samples, the processes, that kind of thing. That must be quite exciting as a scientist, is it? Or do some people, or does it depend on the scientist? Do some people like quite a structured experiment, or or is this broad brush approach, does that might appeal to other people? Yeah. Any thoughts or comments on that?
So this is a kind of a completely different approach than Aimee and I would have previously taken in any of our work, and that we are just being somewhat open to letting people give us the samples that they think are of interest to them, and letting people structure things how they think they might be of value. And then we're going to then just rely on the mass, like just the bulk number of samples got, that then we can use some sort of methods that you'd apply to large data sets. So in particular, we're all familiar with a lot of machine learning or artificial intelligence sort of methodologies that have been employed recently, which sort of seem to be able to take maybe sort of quite incomplete data sets, but still find patterns inside those data sets that can then be tested. If you think of like large language models, how they seem to be able to create what seems like a very plausible, believable sentence just based on statistics, really, of mining data and being able to figure out what words to say with what to make something that sounds like a plausible sentence. So by taking this approach, we're just thinking like, let's rely on current technologies that are available to us, like bulk DNA sequencing, advances in machine learning, and just sort of find out what happens.
Well,
I think we were always going to sort of need the broad brushstroke, as you mentioned, because we want anybody to be able to participate. I think as soon as you put more and more, I guess, restrictions to make the things more structured, then it starts to eliminate a lot of people, and that's not what we wanted. So we tried to make it as accessible to everybody as possible.
No, that's fascinating and fantastic. And I think communities understand, or individuals, a bit more about what the science stuff is when they can participate and see how they can contribute and what it's trying to achieve. And I think there's lots of technologies out there. Well, the internet itself just makes data and information so more readily available. You do, of course, have to apply your own filter to it, so to make sure that it's not fake news or anything like that. But I think, there is a whole lot of stuff that is far more accessible. So we're kind of getting near the end. Is that right? I'm just looking at Dommy, who's the technician here. But some time for you to talk about anything that I haven't raised that you might want to talk about or mention or plug or...
Well, there's one thing that I like to always mention about this project, that there's a lot of benefit in looking in a different way at what we consider to be waste. We will wash down our cars without thinking about the information that might just be on those vehicles. And I think that our project ties into a lot of these other approaches where we've looked at things like wastewater to look at viral load or pathogen load that's in a community. And in the same way, we can look at this just take this as an opportunity to take something that we would have thrown away and learn from it. So without very much cost, we can all benefit. And also, just to reiterate what we're talking about before, I think there's also a real benefit in allowing people to make scientific decisions in how they collect stuff, and to not dictate or to only dictate to the extent that there's a certain constraint on what Aimee and I can do. So people need to sort of fit inside that, but let people be the sort of scientists and make decisions about where they want to sample and just giving over that control.
Fantastic. Aimee?
I guess the one thing for me would probably be, and Richard, would just be to say thank you to everybody that might be listening that's already been involved. We've really appreciated it, and the support has actually been amazing, far beyond anything that we would have expected or even thought possible.
Fantastic.
It's really been so humbling. And to actually engage with people through their comments or their requests or anything like that. It's really been wonderful. And I guess just for anybody that wants to be involved, we have some kits still to get rid of.
Go to the website. Yeah. Thank you very much.
Yes, and to anybody that lives rurally that might not be located in one of those regions, like please reach out. We'd be more than happy to make something work. We want everybody to be that's interested.
Fantastic. Thank you very much for your time. It's been lovely chatting with you.
You've been listening to the OI podcast, the shout out for the arts, sciences and Mataranga Māori.
You can support this podcast by becoming a member of the OI. Check out the Otago Institute for Arts and Sciences on Facebook or our website.
If you'd like to listen again to this episode or others from the series, look for the OI podcast on ORFM's website, oar.org.nz.
Brought to you by the Otago Institute for Arts and Sciences, the Otago branch of the Royal Society of New Zealand.
This podcast was produced by ORFM Dunedin with support from New Zealand On the Air.